Thank you, David. Yeah, hi, I'm Sarah Stone. I'm the Executive Director for the eScience Institute, as Dave said. And I just wanted to, I know Dave mentioned that he has introduced the incubator, the Winter Incubator program to this group in the past, but we're going to have the pleasure of seeing several of the incubator participants over the next few weeks in the community seminar. The Winter Incubator, just to remind you, is a program that we run during winter quarter for over about a 10-week period. The projects that are selected for Winter Incubator come from a call for proposals that goes out late in fall quarter, typically around middle of November to the end of November. And that's an opportunity for researchers at the University of Washington to apply to work one-on-one with one of our data scientists at the eScience Institute. And it's really working shoulder to shoulder, 2 days a week, on a kind of tractable data science challenge in their research. We frequently have graduate students, research scientists, postdoctoral fellows, and faculty participate in the Winter Incubator. A typical refrain, I don't know, we'll see if Charles says this, but a typical refrain is, I got more done during 10 weeks than I would have gotten done in a year or even 2 years. And so, for the right kind of challenge, this is a great opportunity. So I would encourage all of you, if this seems like it might be relevant for your research, to have this opportunity to work with the data science in an intense way, to submit an application or respond to the call for proposals. It's a pretty lightweight call for proposals. It's really just a couple of pages. And then we may have some follow-up questions as we move into selecting the projects that get chosen. So this past quarter, so during winter quarter, we ran this program. The participants, so it's the individual who submitted the proposal is paired with one of our data scientists who's excited to work on that project. They work together generally on Tuesdays and Thursdays for the duration of winter quarter. And this past winter, we had 7 projects that were selected. And again, so you'll hear from most of them over the next few weeks. We'll get rolling today with Charles's presentation. I guess the other thing I wanted to say, if you have any questions about this program, please feel free to reach out to me. You can find me on the eScience website or reach out to Dave and he can give you my contact information. Glad to share what this program is like, what kinds of projects we typically take on, and point you to the available resources. So now I'd like, without further ado, to take the opportunity to introduce Charles Zhou. So Charles is a staff research scientist at UW-NAEP, which is the Neurobiology of Addiction, Pain, and Emotion Center. He worked over the Winter Incubator with eScience data scientist Ariel Rokem. And he's going to be talking today about identifying reward-related spatio-temporal neural maps using principal component analysis. Let's see, feel free, we'll save the questions to the end, and feel free to indicate on Zoom if you have a question, and we'd be happy to field those at the conclusion of the presentation. So thank you, Charles.
Yeah, thanks so much for the introduction. So first I want to say that thanks for everyone for being here, attending and hearing about the work that I've been doing throughout the incubator. And then just to follow up on what Sarah was saying is that I think that the incubator program, or I know the incubator program has been top-notch and spectacular. As Sarah said, I worked with Ariel Rokem, and I've learned so much about the conceptual pieces of what I've done, and then also learned a lot about the increased my coding skills as well. So thanks also to the eScience Institute for putting together such a great program. And then also wanted to thank the scientists that I work with at the NAEP Center. So the data that I'm showing you that I've analyzed have been collected by others at the center, and they've basically done the majority of the work. So with that, I'll talk about, here's my talk. So one of the main focus points of the research center that I work for is understanding how different brain activity patterns underlie and give rise to addiction states. And I think everyone knows here that there's a large impact of drug addiction on society and economic resources and infrastructure across the world and here in the United States. Here, these are plots showing the number of overdose deaths as well as the costs for the opioid epidemic as a function of years, and that number is still increasing quite a bit. So accordingly, it's important for us to understand, specifically for our center, how the brain is able to associate these large rewards that users have when consuming these drugs, and how that link comes about to the different cues that are in the environment. So this is particularly important for drug relapse here. For the example is that someone who is undergoing relapse after a period of abstinence might see like a needle or different paraphernalia or auditory or visual cues that they had previously associated with these rewards and might trigger the relapse. And so understanding this link between this learned association between the cue and the reward could be particularly important for preventing or treating drug relapse. So one way to study this cue reward relationship in this kind of a simple animal model is through a behavioral task, where basically place a lick port in front of a mouse that delivers some sort of liquid, and then across the course of a session, while we're doing brain recordings and recording what the animal is doing, we have a series of randomized trials. In some trials, an auditory tone is played, which is called as CS+. This is essentially the cue that I was talking about. And shortly thereafter, there's a sucrose, or could be water reward, that is delivered. And this is the reward that I was talking about. And then other trials, which serve as control for comparison against, there's a different tone of a different, let's say, frequency. This is called the CS minus, but we'll just call it kind of like unrewarded trials. And there's no reward that is delivered. So during this all, it's important for us to record brain activity during these the behavioral tasks. So the way that we do this is a method called two-photon microscopy, which involves this beautiful piece of equipment. Basically, we place the animal underneath this structure, underneath what's called the objective, and it's able to do the behavioral task that I outlined previously. And the kind of take home here is that this recording method is a little bit similar to magnetic resonance imaging, but think of that on even higher resolution scale. So we can, instead of just recording large-scale brain regions, we can actually see individual neurons. So the data take the form of kind of 3 dimensions a movie, if you will, where we have X, Y space for like the pixels, and then time is frames that you'll see. So you'll notice that the pixels that have that are bright and have high values correspond to neurons that are activating. And so the scale that you see here is on the order of hundreds of microns, so pretty high resolution here. So there's one step of pre-processing I need to do before any further analysis is done, and that is relating these videos to the behavior. And so what I do is, if you remember, across the session there are all these different trials, some that are rewarded, some that are not rewarded. And what I do is, for each trial, I take out a snippet of 4 seconds surrounding the cue and the reward delivery, and then for the downstream analyses, I will do trial averaging, either across trials, across conditions, or averaging across trials within a condition. And so these data are pretty high dimensional. One of the axes is pixels, right? The kind of the spatial information, and that axis has 512 but times 512 number of pixels. So it's that large data can be difficult to work with. So one analysis, or the analysis that I do to make that data more workable, and then also the main analysis that I'm doing is principal component analysis to reduce that dimensionality. And so this method, very briefly, allows for the facilitation of the reduction of the number of features in a data set by finding the combination of features, or in this case pixels, with shared variance. Or in other words, this method allows for me to find the pixels that have common temporal dynamics across or activity profiles across time. And so what I do is I turn this 3-dimensional array that has x, y, and time into a 2D array, which still retains time after I've averaged across trials, across conditions. But then the other dimension is vectorized pixels. So it's combining the x and y pixels. So you can just think of if I just took all these rows and then just concatenated them back to back. And so then what I do is I perform principal component analysis across pixels. And the output of this are components that basically are linear combinations of the original pixels here. So this is one of the components that I'm showing, that you can see that the pixels here are weighted, in that the original pixels that we see here, the ones that are colored in red, contribute strongly to this particular component. And we get multiple of these components where the activity profiles across all these pixels that are highlighted have similar activity, similar profiles. And then also for each of these components, we get the time series across the trial. And I'll get into that a little bit more into the talk. So the first thing that I wanted to do was to examine how much variance explained by each of these components. So how much each component's, or how much of the variability in the activity does each of these components explain? And this is the way that I am able to reduce the amount of features that explain the data. And so what I did was I asked for the number of components that explain 90% of the variability in the data. And I found that there were 18 of these components, and that perhaps the first two explain the most amount of variability, and then potentially the third one as well. And then so what I did next was examine the kind of spatial maps, the pixels that contribute to each component. So for more, I guess, in detail, these are basically the eigenvectors, but reshaped into the shape of the original XY coordinate space. And what you can see here is that these components seem to have profiles. The pixels that contribute strongly to these components have profiles that do correspond to kind of the biological structure. So you can see that the highlighted pixels do look like the neurons that we saw that were activating. So that was a good kind of sanity check. And then I also wanted to look at the other principal components. These are not as defined, but that's probably because with this method we get some unresolved neural activity that can be blurry. So that's expected. So the next thing that I wanted to examine was to look at the activity time series across these top three components. And so what I did was I plotted each of the activity time series of these components against each other into this 3D space. And so what this space means is that, let's say if we just take this principal component, this first principal component, if we see data points or these lines move in, let's say, the positive direction in PC0, this means that the pixels that were highlighted here show high activity. So it kind of gives you a sense of which groups of neurons are activating at a given time point. Another thing is that I have the two conditions of trial averaged two conditions plotted into this space. The red to yellow indicate the unrewarded CS minus trials, and the blue to green are the rewarded CS plus trials. And then further, remember that this is the trials have time as a part of the feature. So time is encoded in this transition from darker to lighter colors. So for the unrewarded, we have the red indicates the time points before the cue or anything was presented to the animal, and then the yellow and then the green indicates when there was shortly after the animal had consumed the reward if it was delivered. So now let me explain this a little bit now. So we can see that at the beginning of the trial, the neural activity looks very similar between the rewarded and unrewarded trials. But then as we go across time, when the animal is able to drink the reward in the rewarded trial, then the neural activity starts to diverge. And that's can be seen in a better format when I rotate the plot. So you can see this divergence happens mostly in principal component 0, which is what we'd expect. So, and then now, what I was also interested in is in the previous plot, we saw I plotted trial average across all trials in the condition. But actually, in this data set, we have 50 trials for each condition. So another question that I could ask is, well, how does learning occur, or how does learning, is there a learning component across the trials in a given in a session? And so what I did was I grouped trials in groups of 10, and then averaged those trials and then plotted them into this space. And basically the take-home message here is, indeed, there does seem to be this evolution of activity with respect to principal component 0 across these trials, across sessions. So again, if I rotate this space, we can see that here the lighter alpha trials, group trials, indicate the trials that occurred at the beginning of the session, and then the darker at the later points of the session. And you can see this gradual evolution or increase in activity across this principal component 0 across sessions. So basically the take home here is that there is a differentiation of rewarded and unrewarded conditions across principal component 0. So the neurons, the pixels that correspond to these neurons in principal component 0 seem to have higher activity for the rewarded trials as well as across the session, the trials across the session. So another way to visualize the data that I just showed you was to look at the activity time series for each component. And so here you can see the activity across time, where time at 0 is when the cue was presented, and that we have the unrewarded trials in red and the rewarded trial condition in blue. And we can see that again for principal component 0, that there's this large increase in activity for principal component 0, but not so much for 1 and 2. That could mean that the pixels or the neurons in principal component 1 and 2, they're still activating, but they could be responding to components of the trial that are not directly related to the reward, such as the animal licking or detecting an auditory tone. And then finally, what I wanted to do was to examine the actual physical maps, the activity maps at different time points when combining these three principal components at these different time points. So what I did was I multiplied the eigenvector for the top three principal components with the activity time series for those components, and then took the average across time for the period of time before the cue or anything that was delivered to the animal, and then also after reward delivery. So when we started to see that divergence in activity. And then the first column here is the unrewarded trials, and then the second are the rewarded trials. And you can see that clearly that at this later time point when the animal had received the reward, that there is these neurons that show heightened activity. So again, the final take home that I have for you is that this method of analyzing these video data of neural activity allows for us to figure out which ensembles of neurons are heavily related to this learning of this cue to a reward. And this would allow us, we have tools in the lab and the center now to be able to stimulate specific neurons. And because now we know which specific neurons could be related to learning this cue and reward relationship, we can activate or suppress them to see if there are differences or changes in behavior during drug relapse or drug learning. So with that, I'd like to thank, again, many thanks to the eScience Institute, in particular, Ariel Rokam and Brenna Hazelton, the two mentors that I've learned so much from throughout, as well as the incubator fellows. Vijay Namudiri is a postdoc who I work with who collected all the data, and then the NAPE Center as a whole, and my two mentors there, Gareth Seaver and Michael Brukas, as well as our funding sources. And I'm open to taking any questions. Thank you.
Thank you, Charles. So I see a question from Ariel. Reminder, if you want to ask a question under the participants list, please raise your hand. Ariel, do you have a question?
Yeah, so my question is, so the activity, this thing that looks like learning, the question is, how do we design an analysis that will tell us that that really is learning, as distinguished from, I don't know, other kinds of things that could affect the animal, like they might be getting tired and things of that sort. What kind of stuff could we do here? I'm sort of brainstorming to think about what is kind of the next steps in this.
Yeah, that's a great question. Yeah, we could, you could argue, and that's a totally valid argument, that as the session goes on, the animal might either be more aroused, or probably in this case less aroused, and maybe there's like a negative correlation between arousal and activity. One way to, I understand that is just one kind of explanation for this activity, but just focusing on that, one thing that we could do is record the pupil diameter, because we know that the pupil diameter, the size of it correlates with how aroused animals can be, and so that what we can try to do is look at if there is indeed a correlation between the pupil diameter size and the activity that we see, I guess, in these both in the raw data and then also that we see here in the reduced dimensionality state. And then other things that we could do to kind of further pinpoint if it really is learning is it's kind of further touching on what my take-home message is that we do have ways to basically suppress the activity of these neurons. So what we could do is in a targeted way. So we could basically suppress the activity of these specific neurons that I highlighted, and then see if there is an impairment in learning this association between the cue and the reward, which would be, I think, in the case of drug addiction, would be good, because it would basically decouple the positive reward feelings that, like, let's say the animal has with a certain cue, and so that it could prevent relapse. But yeah, that's a great question, and thank you.
Awesome. Thank you, Charles. So April Craig has a question. April?
Hi, great job, Charles. I was wondering, so in your experiment, you used a sound as a stimulus. I was wondering if you think the results would differ in any way if you used a visual stimulus or some other type of stimulus, or multiple at the same time.
Yeah, that's a good question. I don't have an exact answer for you for there, but I think I would hypothesize that it wouldn't make a huge difference. One is because there's been a lot of research showing that this particular, well, one thing that I didn't highlight is where I'm actually, these recordings are happening in, but it's in the orbital frontal cortex. So it's part of the frontal cortex. The important thing there is that it's not where, it's a region that doesn't quite, it doesn't differentiate different stimuli, rather more of these abstract reward learning. So because of that, that brain region might be more interested in broadly how the reward connects to any sort of stimulus.
I see.
But that would be definitely an interesting set of experiments to look at.
Thank you.
Thank you, April. Stuart Graham.
Hi, Charles. That was a great talk. Thanks for sharing. I had a question about how the divergence between the control and the rewarded trials. If I'm understanding correctly, it seems like the brain activity of the two trials diverges only a little bit at the beginning, which I presume is when the sound is being played, but then diverges a lot towards the end, which I assume is after the reward is given. So to me, if I'm understanding this correctly, that suggests that the divergence is mostly in the response to the reward rather than the response to the cue for the reward. And if I'm interpreting that correctly, is that a pattern that's surprising or interesting, or?
Yeah, that's a great question. And I don't have a great answer to that, but you are correct in that the largest divergence in the activity is happening during that rewarded time, and not so much for the cue. I think what we would need to do, so one thing that I will say is that the data that I use here have been published, and the paper that was published has evidence that these neurons are linking the cue and the reward together. But I think that one set of experiments that or I could look into if they have done this already, is to see how the activity changes when you take the cue out. Yeah.
Thank you.
Thank you for the great question.
Thank you. Any more questions? Well, I want to thank our speaker again, Charles. It's a little hard for us all to clap in the traditional way, but we do have the clap icon, so everybody can use the clap icon.
Yeah, thanks everyone for attending and hearing about the work.
Thank you. It was a really great talk, and I appreciate everybody being willing to adapt to the new way that we're living now. Sarah, do you have any closing thoughts that you want to add?
No, I really enjoyed the talk, Charles, and thank you for coming and presenting in this forum. we'll have another speaker next week from our incubator session and over the next couple of weeks. So hopefully you guys will be, enjoy hearing these very diverse talks, is very much in line with what you normally hear in the community seminar, getting to hear about data science challenges from very diverse perspectives and with a great breadth of disciplinary applications. So thank you, Charles.
Yeah, thank you.
Awesome.
Thanks for everyone.
Thank you, Sarah. All right. Well, we're ending a little early today, and I know everybody's happy about that. So enjoy the sun and have a great afternoon.